Hello friends, welcome to Scientech Biology. In this video, we are going to see the different phases of cell cycle and how the cell cycle is regulated. So let's begin this video. A cell cycle is a series of events that takes place in a cell as it grows and divides. A cell spends most of its time in what is called interphase, and during this time, it grows, replicates its chromosomes, and prepares for the cell division. The two main events occur in each cell division. The first is the doubling of genome in the synthesis phase, also called S phase, and the second is halving of genome during mitosis phase. The eukaryotic cell cycle completes in two phases, the interphase and M phase. Interphase is the preparatory phase of the cell cycle which leads to the doubling of genome. The interphase is further divided into G1 or post-mitotic gap phase, S phase or synthesis phase, and G2 or pre-mitotic gap phase. M phase, which is a real phase of division, is itself composed of two tightly coupled processes: karyokinesis, in which the cell's nucleus divides, and the cytokinesis, in which the cell's cytoplasm divides, forming two daughter cells. The mitotic phase is divided into prophase, metaphase, anaphase, and telophase. In M phase, the genome becomes half. Now let's see in detail the different phases of cell cycle. In G1 phase, the different enzyme which is needed for the DNA replication are synthesized, as well as ATP and RNA molecule also synthesized for the transcription and translation process. So many things are synthesized in G1 phase, so the size of the cell increases. In G1 phase, a cell has three options: either to continue cell cycle and enters S phase, stop cell cycle and enters G0 phase for undergoing differentiation, or becomes arrested in G1 phase, hence it may enter G0 phase or re-enter the cell cycle. Cells of liver, kidney and neuron, after remaining in G1 phase for some time, comes out of the cell cycle and enters to G0 phase, also known as quiescent phase. In quiescent phase, the cell division stop, but the other activity within the cell remain continue. After the completion of G1 phase, the cell cycle transitions into S phase. DNA replication is the main event that occurs during S phase. Some kinds of histone protein is also synthesized in S phase. Usually cells takes 5 to 6 hours to complete S phase. After S phase, the cell enters into G2 phase, which is shorter, lasting only 3 to 4 hours. In G2 phase, the following events occur: the tubulin protein synthesized, which is required for spindle formation. For plasma membrane formation, certain proteins are also synthesized. ATP synthesis and RNA synthesis also occur during G2 phase. If you enjoy my video, be sure to hit the subscribe button and press the bell icon to make sure you get notified when I upload a new video. The next stage is M phase. M stands for mitosis. This is where the cell actually partitions the two copies of the genetic material into the two daughter cells. In prophase of mitosis, chromatid coiling, spindle formation, and disintegration of nuclear membrane occurs. In metaphase, the chromosomes oriented at equatorial plane. In anaphase, the sister chromatids separate from each other and moves towards the opposite poles. And in telophase, which is the last phase of mitosis, the chromosome reach the poles and nuclear membrane reforms. Telophase is followed by cytokinesis, or the division of the cytoplasm into two daughter cells. Activation of each phase of the cell cycle is dependent on the proper progression and completion of the previous one. The deciding point is called checkpoint. The checkpoint ensures the proper progression of the cell cycle. The whole cell cycle is controlled by the three checkpoints, which are the G1 checkpoint at G1 and S transition, the G2 checkpoint at G2 and M transition, and the M checkpoint or spindle checkpoint at the transition from metaphase to anaphase. The G1 checkpoint is the main decision point for a cell, where it decides whether the cell will divide or remain suspended. The G1 checkpoint checks all the preparation needed for the cell to enter into the S phase. The next checkpoint is G2 checkpoint before M phase, which ensures the proper DNA replication and checks whether M phase cyclin and CDK complexes is activated to initiate mitosis. The third checkpoint is at the transition of metaphase and anaphase. Here the checkpoint examines whether all the sister chromatids are correctly attached to the spindle microtubules or not. In addition to the checkpoint, there are two groups of intracellular molecules that regulate cell cycle. The two groups of protein called cyclins and cyclin-dependent kinase are responsible for the progress of cell through the various checkpoints. Hope you enjoyed this video. Thanks for watching.